Hello, and welcome to the week 8 of the live interactive session of the Neural Science for Engineers. The course instructor for this course is Professor Vikas V from NIMHANS Bangalore, and I am the PMRF teaching assistant Vikas Pandey from IIT Jodhpur. This is a brief about me. For more details, you can visit my portfolio. The schedule for this class remains the same, that's Tuesday 6 p.m. to 8 p.m. And this is the Meet link. All the recordings are available over the YouTube channel. For any concerns or questions related to the tutorials or these live interactive sessions, you can even mail me over my email. So, as we have been doing from the last 2-3 weeks, that we are first discussing the summary of the lectures, then we are going towards the question. So, let's first discuss this a summary in short, though you should see the lectures for the complete details to understand uh, how the brain works and how engineers can solve many problems related to brains, neural science, and all. Okay, so now the lecture is mostly focusing over visual processing. Okay, so visual processing, as the name suggests, it involves starting from photon reception to the higher level of cognition. Like uh, from the point the photon, that's the light, enters our eyes till understanding what is going on, how we understand that, like we process that data and everything, that is the part of the visual processing. Okay, we have seen already that cones, like there are two type of cells, cones and rods. So cones in the retina are stimulated by photons of some specific wavelength. That means for say for red, some specific set of cone cells will be activated. For blue light, some specific uh, cone cells will be activated. For green, some specific cone cells will be activated. Okay, so how we uh, can say that this is the mechanism, and as we know that there are three primary colors that make all the colors of light. Okay, so very similar to that, there are three different receptors or cone cells in our retina. Then the initial contrast detection and basic image processing occurs in the retina itself before going towards brain, and that is done by the rod cells. Right? Cone cells. How I used to remember was cones for color and rods for contrast. Okay, so that will make it easier for you to remember it. Okay, then comes the part where there is some intermediate processing and pre-processing. So intermediate processing involves spatial integration, that is the perception of space, that means dimensions, you can say, and coding in the lateral genital body. Okay, this lateral genital body acts as a relay station. So that is the part where the integration of data occurs from our eyes before transmitting it to the visual cortex for further analysis. So if we see it like a computer system, okay, so this is a sense. Eyes are a sensor. They detect the data. Some part is processed here itself because as it is, or when we talk about sensors, say it is receiving in analog form and it is some digital conversion or something like that is happening. So very similar to that, our eyes do like receive in visual information in form of light, photons. Then in the retina itself, some colors are separated because they will have a different path since different cones are detecting them. Then lateral genicutal body, that will integrate this data and transmit it to visual cortex. The pre-processing tasks such as color contrast understanding and color fine-tuning are performed in the retina itself, that I said. So light is coming, some pre-processing, that is contrast and color understanding, is happening there. We are not understanding, you can say that and a colored image and a contrast containing image is formed. You can say that rasterization kind of thing is happening. Okay, then it is being integrated and transmitted to the visual cortex for the further understanding. Then comes the higher level cognitive function. Beyond basic processing, high level function include object recognition, emotional balance attachment, and memory integration. You see something, you remember something. Oh, this is related to that particular thing. Or if you see someone and from your memory you check that, okay, I have met that person. This is something like higher level cognitive function. So, object recognition, that involves categorization and association with the past experiences. Then is emotional balance, like seeing something you become very happy, or seeing like a child, like when a child sees something that makes him happy. Okay, how he does that? Because he attach, like he meet say he meets his uncle after some time, so he will feel happy because uh, he has some past experiences. He or she the must have some past experience where, like, the face of the person is related to someone with whom they have some happy memories or e- good emotional connect, and exactly the opposite for bad emotional connects. Okay, then memory integration seamlessly merges visual information with contextual information. So it is not just the vision, along with that some emotions, like something is happening. You might have experienced this, that I used to wonder earlier how this happens, that when we see, like, it used to happen that when we see some picture related to a place that we might have visited, and what is there, that there is some like there is some say there is some temperature difference, was there was some it was a cold weather when you took that picture, and there is some emotional connect there, as well as there was some specific smell that you remember in your memory. When you see that picture, those things you can feel a bit. So this is how this like, you can say that picture, it is very different from how the pictures are being saved in our uh, hard disk and phones and computers, where only the pictorial information is there in our mind. Along with that, there are other things also, like emotion and I can say smell, and these things, they all are stored with that memory. Okay, then comes the control of eye movements. So we we should not assume these eye as just cameras that take passive pictures, but they actually process visual information in the brain also. Okay, so uh, there are four eye movement mechanisms: SCADs, that is rapid unconscious movement shifting fixation point within a visual scene. So you are seeing something, but in that frame or scene itself, you unconsciously, your body eye will move a bit. So if you see some person, uh, that person's eye, like, I will see changing the positions, that is schedules. Then is the pursuit, tracking moving objects to maintain fixation. So if you are seeing a car that is moving, you will not have to put any effort, and you will see that actually your eye continues the gaze, and you keep seeing the car. Okay, that movement is pursuit. Then is vergence, that is adjusting the convergence to focus on objects at different distance. So something like you are going uh, on a vehicle, and you see two not speed breakers, they are like hurdle kind of structures moving at some distance, like where you have to move zigzag. Okay, so in this case, how you see these two things like separating each other at some distance, that is the vergence. And then is the vestibulo-ocular reflex that stabilizes the gaze during head movement. So you, if your head is moving like this, like I am moving, but still I am seeing towards the camera. And how this is being done? This is being done by the vestibulo-ocular reflex. The reflex is very similar to the reflex that we studied in our spinal cord part, right? Where some reflexes were there. Here, the reflex is this is due to the uh, you can say there is a gyroscope kind of structure in our eyes that take care like how we are moving, and so the reflex occurs that even if my head is moving like this, the eye remains the same. Okay, so how these eye movements are actually done? The specific muscles that are controlled by cranial nerve mediate the eye movement. Okay, the synchronization of left and right eye occurs through direct neurological connection. So, since it is very near to the brain, it is directly connected to the brain through without you can say without any intermediary step part, and that facilitates very fast movements. Okay, midbrain nuclei are crucial in coordinating the eye movement and gaze control. Okay, eye movement serves the purpose of perceiving different objects and threats and maintaining balance. Okay, various eye movements mechanism ensure visual tracking, focus adjustment, gaze stabilization. So why we are doing this? Because understanding the neurological basis enhances knowledge of vision processing and perception, and that can help us make some improvement. Some people suffer from some problems. We can diagnose and we can correct those, as well as under if we have a good understanding of the visual system, we will be able to devise things that will help people who don't have this visual system. So using electronic tools, at least if it cannot be a replacement of say 10% of the vision, at least it will restore say 50-70% of the vision. Like the person will be able to visualize things. So that is why this part is necessary to understand. Then comes the second part of this week, that is the COMSOL multiphysics for medical devices. So COMSOL is you can say a numerical tool for various. It is a numerical tool calculate. You can say a very high level calculator that have equations and solving some numerical data. A lot of numerical data can be solved simultaneously. It can be handled through equations and all. So COMSOL is that. Okay. Why is it needed in our case? It is needed for simulating medical devices. Why? Uh, why we need to simulate medical devices? Why don't we directly test on humans and primates and all? Okay, that is the way that was doing. Because in this case, we will have a faster time to market. Trial and errors will be lesser. Okay, trials and errors will be done in the simulation. Okay, then we will also understand the physical phenomena behind this. Many times it happens that something is working, but why is it working? That again requires a different study. Okay, then as I said, we will reduce the number of prototypes. So we will come towards like we will narrow down towards some specific ways that will work, and so we will have lesser number of prototypes to try. We don't need to go for more trial and errors in physical world. Okay, so and okay, this I think I have covered in this only. So what is the present challenge that is there in case of biomedical engineering? That is the the tissue properties that we assume in physics or any other simulation. that we assume to be uniform. Okay, but it is not so in case of you can say in real scenario it is not like that. There are variation. As you can say that same type of tissue, two tissue kept together will have different properties, electrical as well as other properties. Okay, there will be like we can take some tolerance. Fine. Then tissue damage modeling is still a problem. How how much tissue will be damaged and all. Again, like the physical phenomena we study in idealized environment. We take a lot of ideal conditions and all that cannot directly be kept in case of simulations. Like in simulations that is kept, in case of real life scenario it will have some differences. Okay, so what is the solution at present? That we homogenize everything, like we take the average or something like that, and then we homogenize. Second is we cannot model each and every tissue, each and every part, small, so we take a mathematical model here, like many times in our school days or college, even in starting of the college, many students used to say that why we are studying much of mathematics, what is the need of it in future except for calculating our grocery. But mathematics, mathematical models are the foundation of almost all the simulation tools. They can reduce the effort a lot. Like you have a lot of data, you have a lot of points. Using mathematical models, we can create an equation having very less number of variables, and we can replicate that in simulations. Okay, and another solution is incorporating the blood flow dynamics in simulation. So when we incorporate the blood flow dynamics, it replicates a bit of the real life, uh, real scenario. Okay, then comes the electrical simulation of brain. That is again the part of the COMSOL simulation itself. And why is it important? Because for biomedical application, it is something that we use. Okay, what are the steps? The step is first we choose the simulation type, that is that can be DC, frequency, or time domain. Okay, then we model the setup using COMSOL model wizard. Okay, then apply physical conditions of the material, then mesh the geometry for solver accuracy. So if we create a structure, say like this, and we say that this is epithelial tissues, some other tissues, okay, and like that. Then what we do is that we first say that what is the model for this, mathematical model for it. We define the mathematical model for that. This, all three we have done. Now we cannot have something that will do this for infinite number. Like when we talk about analog world, like the real world, there is no specific point or specific tissue or something. Like we cannot do for individual ones. That will make it very complex, very time consuming. So we do a messing of the geometry. So how this is, you can say that this is that. Let me change the color. Mm. So this is patterned into mesh. Mesh is simply net kind of structure. Okay. So what this mesh does is that it reduces the number of our equation, and minded that it is not always necessary to have a uniform mesh. In fact, mostly areas where we have more concerns, like say on the boundary areas, on the junctions, we want uh, better understanding, and we are expecting that some changes will be more there. There we take more mesh, like that will be more dense. And what it does is that if we take, let's say, these are the mesh points. Okay, so the equations are not solved at each and every point. Instead of that, they are generally solved at these intersections only. The properties at these intersections will be solved and analyzed, and then you can easily expand, assume that these are for the rest area of this. Like average of this will give the this whole area of the parameters of the whole area. Okay, that is something that we do for the uh, in case of simulation. Okay, so how then the post, once this is done, there will be some result. So we cannot see the equations, like we will not be able to understand very well those equations and all. So what we generally do is that we visualize it in terms of electric potential and current flow. We try to understand that how the current or the charge profile is there, and how the current is flowing through it. Then the second is animation, that is the phase dependent animation for complete understanding. So we keep it, and like we see that how the console will show that how with different colors, how the charge flow is there, how potential is forming, and all these things. Okay, so after this, let's go towards some problems related to the topics that we have covered in the week. Okay, so let's come with the first question, that is, what happens when light stimulates a cone in the visual pathway? So there is a cone in our visual pathway that is there in the retina. So what will happen when light reaches there or stimulates that? There will be bipolar cell inhibition, reduction in reception activity, ganglion cell hyperpolarization, or increased activity in the horizontal cell. Yes. Okay. So what happens is that when light stimulates a cone, it reduces the receptor activity. That actually increases the output activity of the ganglion cell. So answer B will be the correct answer. That is reduction in the receptor activity. Okay. Now let's come to problem 2. Which term describes the phenomena where one photon stimulates one ganglion cell to be on while an adjacent cell is off? Is it lateral inhibition, multisynaptic transmission, hyperpolarization cascade, or receptive field modulation? So it is lateral inhibition. This refers to the process by which activation of one neuron inhibits the activity of the adjacent neuron. In visual processing, lateral inhibition results in one ganglion cell being activated while an adjacent one is inhibited. Like cone, once it is stimulated, the adjacent ones it will off. Okay, that means the related ganglion cell will only be active. Okay. What is the significance of layer 1, 4, and 6 in the lateral geniculate nucleus? They process ipsilateral visual data. They process contralateral visual data. They are responsible for color perception, or they receive input from the optic tract. The layer 1, 4, and 6 of the LGN receive contralateral visual data, while layer 2, 3, and 5 process ipsilateral data. So in our case, the answer will be B. That is, they process contralateral visual data. In the visual cortex, what function do bulbs serve? Detection of color, orientation preference, depth perception, or motion detection? Blobs within the visual cortex are involved in detecting and processing the color information. So the correct answer is A, that is detection of the color. Binocular disparity. Bi, whenever we talk about bi, that means two. So binocular dispolarity refers to difference in receptor sensitivity between the eyes, the distance between the eyes, difference in focal length between the eyes, that is one eye having different focal length, other eye different focal length, the difference in the position of image on the retina of the two eyes. So what can be the answer? So the answer is the difference in the position of images on the retinas of the two eyes. This phenomenon, that is the binocular disparity, that gives us the spatial understanding. The same thing, like say I'm seeing this pen. If I am seeing it with the left eye, the image that is formed is somewhat like this part. Okay. When I am seeing from this eye, it is having a separate image. You can say that if you can see that if you just see it with one eye, you will be able to see one part of the image. When you see with another eye, you will be able to see another part of image. This gives us, this difference actually is something that gives us the understanding of the depth. Okay. uh, if you can try with try seeing two first like separate two things, uh, like you can say that okay, I have these two things, I have these two at the same level. If I move this forward and this backward, okay, if I am seeing with two eyes, I will be able to make out which is near me, which is far away from me. But if I see it with only one eye closed, that is very difficult to make because uh, we will not have this binocular disparity. Okay. What role do neurons with specific orientation play in visual processing? They detect changes in brightness, they encode path depth information, they respond to specific orientation of visual stimuli, they are involved in color discrimination. So, neurons with specific orientation preferences are responsible for detecting and responding to particular orientation of visual stimuli, such as lines or edges. Okay, so they respond to specific orientation of visual stimuli. Hello.
Hello, sir. Hello, sir.
Yeah, how are
Sorry
you?
for the late, sir, because of
No, not an
some
issue.
urgent meeting.
Okay, not an issue. Okay, so let's the first part of the lecture you will find in on the YouTube. This let's continue from here. I have discussed the summary mostly today. Like in this week, we have mostly covered the visual uh, visual part, how the vision processing and this visual processing occurs, and the second part is COMSOL multi-physics simulation. Okay, so let's move forward with some more questions. Fine.
Yes, sir. Yes, sir.
Yeah. So, what is the purpose of eye movements? To regulate the diameter of the pupil, to perceive different objects and threats in the environment, to maintain balance and equilibrium, to enhance auditory perception. Yes, any answers?
B. To perceive
B.
different objects and threats in the
Objects and
environment.
threats in the environment. The eye movements perceive different object and potential threat in the environment by scanning the surrounding and shifting from one point of fixation to other. So these eye movements that we see in our case, that is to perceive different objects as well as threats in the environment. Okay. Which of the following eye movements is primarily unconscious and involves rapid short amplitude movements? So the part that we are not able to uh, control. Pursuit,
Cats.
vergence,
Second.
scats, vestibular, ocular reflex. So it is scats. Like
Second.
yeah, in the summary part, I get this right. So scats are rapid, mostly unconscious eye movement characterized by short amplitude movements within a visual scene. So we are seeing something, we are not trying to move our focus, but yet there will be some short moments. They are essential for shifting the point of fixation quickly and efficiently. Okay. So, what is the purpose of pursuing eye movements? To focus on stationary objects, to maintain balance during head movements, to track moving object, to adjust the diameter of the pupil.
One second, sir. What is the purpose of eye movement? To focus on
I think there is spelling
remove, sir.
mistake.
Remove mode screen.
Yeah, no, this is actually
This is
autocorrect.
actually
Yeah,
what we are doing means
uh,
it has to reach the forevia. For that purposing, pursuing eye movement is to
yeah. No,
focus on
pursuing it is pursuit eye moment.
yeah.
Like it has got auto-corrected, like smart computers do this. So uh, I had written
I think
pursuit and
both A and B and C also same, sir. Purpose of
no,
eye movement.
there are four.
Okay. Actually, uh, focus on stationary object. A.
There are four types of eye movement, right? So I think that part I can. So yeah.
Four are there,
So
sir. Saccades is
yeah,
there, then
the
pursuit,
saccades,
virgin,
pursuit,
vestibule.
vergence, and vestibule. So
Hmm.
saccades is the unconscious, rapid unconscious movement. Pursuit,
Hmm.
that is to track moving object to maintain fixation. Vergence is adjusting convergence to focus on different
Yeah.
objects at different distances, where we
Yes, sir.
get this spatial and vestibulocular reflex. That is that when we move our head and yet eye will keep focused on that object. That is the vestibulocular reflex. This reflex is very similar to the reflex we studied in case of spinal reflexes and all. Right. So based
What role? Okay, specific
on
role.
yeah, on this we have covered. Hmm. So it is to track moving object. Track and maintain fixation on moving object within the visual field. Like you are seeing a car that is moving forward. Okay. So the moment that is that keep focused on that, like the car starts from here, you are seeing that, and you will see the car moves and your vision moves along with that. You will be focused on the car. Okay, moving car I am talking about. Okay, so uh, enabling this enables continuous visual tracking without needing the head movement. Like we don't need, like there is something coming from, like in my visual plane you can say something comes, I see there, and then it goes like this. Without head movement, my eyes will move with it. That is the pursuit eye movement. Okay. Then is which eye movement mechanism help focus on object at different distances by adjusting the convergence of the eyes. Pursuit, vergence, scadals, or vestibulocular reflex.
Which eye movement mechanism help us focus on object different distance by
Yeah.
adjusting different distance. That is
Like
pursuit. Pursuit only. Vergence is
no,
near the eye.
vergence uh, is
Near
something
the
that
eyes. Near the
that
both the
hmm,
pupils will oppositely moves. That is called vergence.
hmm.
Opposite direction.
That is this is about convergence where you are converging, like two eyes seeing towards one thing.
That is
Okay, how
vergence
it is.
only.
Yeah, that is vergence. So
So
in this case, convergence, it will be vergence, no?
okay, okay.
Yeah, where it is converging. That gives us
Converge.
the understanding of this distance. Like I was explaining earlier, like if I have these two objects, right? And if I am seeing with both my eyes, I can understand which is forward and which is backward, right? But if you try to see it with one eye, you cannot make out that which is uh, far away from me and which is near me. This uh, this we are able to do. That is the binocular vision have because we have two eyes. There is if you see something there, this part like the left eye will have a different image, right eye will have a different image, and the convergence of these two images makes us see the have the binocular vision, right? So, what is the function of vestibulo-ocular reflex? To maintain balance during head movement, to stabilize gaze during head movement, to adjust diameter of pupil in response to light, to synchronize eye movement between the left and right eyes.
It is B, sir. To stabilize gaze during head movement.
Gaze during head movement, because as we discussed, that is something that is not making the eye move along with the head. Like the vision focus will remain there. Okay. Then, how are eye movements synchronized between left and right eyes? There is a conscious effort to control each eye independently. There is direct connection between specific eye muscle pairs through change in the diameter of the pupil, through change in through auditory feedback from the vestibular system.
It is D, sir. Through auditory feedback from vestibular system.
Okay, so we are hearing something and then we are changing our eye movement.
No, vestibule system will give the position of the head. As per that, I think it is
It
synchronizing.
is it is synchronizing both, like it is asking left and right eyes. Okay, it is not about movement of head. Okay, that is a different thing. I am here, I am trying to see the camera,
Oh, okay,
my
okay.
eyes
Go.
move there.
Oh,
Okay, then I try to see something here, my eyes in sync move towards that. Okay, how this
then B, yeah.
synchronization is.
B answer is B. Yeah.
Yes, so we have uh, there is are directly connected specific eye muscle pairs. Sorry, I'm saying eye muscle pairs. Okay, that control them. So when I'm moving this on the left, the other eye will automatically move on the left until and unless I am trying to control it forcefully. Okay, this ensures coordination, movement, and gaze stabilization also. So there is a role of this in gaze stabilization, whereas there is a role of the vestibule this vestibular system where we are yeah, like whenever we have head movement, then also we are able to see things without gaze. But along with that, the connection of these two eyes working in tandem, that also makes it possible. Which part of brain is responsible for coordinating eye movements and gaze control? Cerebrum, cerebellum, hippocampus, midbrain, prefrontal cortex.
I think prefrontal cortex only, no? The
Yeah,
cortex only finally it will go.
yeah. In cortex, what happens? Is it controlling the movement of eyes, or is it understanding our visual information?
See, hippocampus is memory.
Hmm.
Midbrain is your emotion. Then cerebellum, cerebellum maybe, sir.
And cerebellum, what does the cerebellum do?
Is
Where
coordinating
is
all muscle movements. Or
where is
maybe
our
I you know
memory located?
cerebellum nowhere it is there, so must be peripheral cortex only. I'm saying first, no, but you are saying it is
Yeah,
a
it's
midbrain.
the
Okay.
midbrain.
Uh,
Okay, that contains the moment. When you are saying about cortex and cerebellum, they contain they do this memory integration and understanding part. But the movement, that is, you can say that mm, if we talk in terms of cameras, though eye is much complex compared to a camera. So the shutter control, when we are talking about this eye gaze and this, so shutter stabilization, like lens stabilization and shutter control kind of work. Okay, Then the processing of the image, that is a different part, and shutter control is a different part, right? Both are not the same part. In case of a camera, if we are seeing a camera, then are they different or they are done by, by the same thing? Are you
Yeah, yes, sir.
understanding
I
my
am here
question?
only. Couldn't the midbrain contains nuclei coordinating eye movements and gaze control.
No, this
Where is
is
the
the same.
normal? That may be in thalamus, huh?
Yeah,
Cranial
like
nerves, ensuring
what
this.
I
Okay.
what I am saying is that, like, if we understand this our visual system, if we think in terms of camera, that is a simpler model of our visual system, right? So uh, nowadays you must have seen that there is something called optical image stabilization. Are you aware of this?
No, sir.
No. In most of the phones nowadays, we have something called optical image stabilization. Okay, so what it does is that, uh, what it earlier phones, they 10 years back, when we used to take video, due to our hand movements, the video will come shaky, right? Nowadays, the same camera, same lens, but when you take the image, there is something that, actually, when you see the video, you will not see the, like, your hand movement, slight variation or slight shaking due to your hand movement, that is gone. Okay, that is being taken by something called as optical image stabilization. Okay, so we can understand that this eye movement coordination is very similar to them. Okay, so there is a separate processing unit, uh, special part kept. If you see the most of the common models of the phones and all, you'll see that they advertise it as it has optical image stabilization that is added, and that is that it will move the lens according to, opposite to your movement. Okay, and this is something that has been copied again from the eye movement. Okay, so uh, they have different, like, and that is not the part of the where storage of this image and video is taking place. So similar to that, in our brain also, these two are separated. The storage and memory creation, that is in cortex and cerebellum, but the control is via the midbrain. Okay, next, let's come to the COMSOL multiphysics parts, where we saw that how the brain is simulated and all. Okay, so what is the primary purpose of using numerical tools like COMSOL? As I discussed earlier, that COMSOL is a numerical tool. We can say a very complex calculator that can take equations and all other things. Okay, so why is it needed for medical device simulation? Is it to replace animal testing completely and directly, like we simulate it and directly go on human trials, or to achieve faster time to market for medical devices, or to eliminate the need for physical prototypes, or to reduce the need for clinical trials on humans? So we don't do trials and we directly use it on humans. So what will be the correct answer? Yes, any answers?
Animal testing completely achieve fast and need of physical water to reduce need for clinical trial. I think Cosmol is multi-physics tools.
So
That
it is
is
simply, let's forget about this COMSOL multiple. Let's say that we are simulating the medical devices
yeah,
before
yeah.
bringing. Uh, so what will this simulation serve?
It is to eliminate the need for physical proto. I couldn't understand. All are look same.
Okay.
But I know the
So
use of Cosmol. This just like
console,
I'm
yeah.
working in CFD, so make
Yeah,
a
yeah.
mesh and do the analysis,
Yes,
multi-physics there.
exactly the
So
same thing. Yeah. So when we do this, we we are not actually replacing the whole process, but we are like we are reducing the physical number of we are not removing the number of overall physical prototype, but we are reducing. Like earlier, say we had to test 10, 20 prototypes. Now you are coming to a position that like these two types will generally work. So we are reducing the number of physical prototypes. And making prototypes is costlier as well as time taking. So in this process, it reduces the time for the market for these devices to be available in the market. So first, do the simulation. Whatever results you get, you understand that these are like less number of prototypes you are ready with. Then you go with the physical prototype. So it is to achieve faster time to market for medical devices. Certainly, animal testing will remain there, ethical clearance will remain there, human trials will be there, but creation of the devices that reduces, that time reduces. Okay, so this is something that industries do, especially uh, especially when there are multiple phases of trial that are involved, like animal testing, primate, like rat testing, primate testing, and then human testing. Okay, so they are able to understand intuitively, like, okay, this physical phenomena is there, so we are we will be able to do it using such models. Okay, trials and errors will be less. Fine. So, what is the one challenge mentioned in biomedical engineering regarding tissue property? Okay, it's heterogeneous tissue properties, uniform tissue properties, homogeneous tissue properties, or predictable tissue properties.
what is the challenge in mentioned biomedical engineering? Heterogeneous tissue property.
Yeah, and why is it so?
Because it is not a normal material, one property will be there. This is a organic material, no different
Yeah, organ. Yeah, and
tissue.
this
It is
is
not
we
obeying
can
Hooke's law or any
yeah
other law.
yeah. So no law is there. Like each tissue, though look similar, though will be of the same type, still be different, right? So this is a challenge in case of simulation in tissue like biomedical engineering compared to our normal material science and all. Okay. So how it is tackled by using a homogenized approach or mathematical model to different to represent different tissue type. Okay, what is the purpose of simulating blood flow within neural tissues? So we we are seeing tissue level, and when then we are simulating the tissue. Along with that, the blood flow within neural tissue is also simulated and mathematically modeled. Why is it so? Is it to understand the composition of blood, to visualize blood flow pattern, to study the behavior of platelets, or to determine the effectiveness of hearing aids?
I think it is B, sir, to visualize blood flow patterns.
Blood flow patterns. And simulating blood flow within neural tissue help visualize blood flow pattern, and this helps us understand the distribution of constituent like red blood cell and platelets, and then we are able to study properties like viscosity and other electrical properties also. So it is not just the tissues alone. So here one extra complexity is being added to our simulation, that is we are simulating the blood flow along with the tissue model and all. What is the primary concern addressed by simulating thermal damage in brain tissue? So, not only this is the problem, another problem is that we are not able to uh, understand that how this tissue will get damaged due to the electric current and all, like we were saying, stimulate. This is simulating, and there we were stimulating using electrodes, right? So, how this brain tissues will respond? We know the electrical impulse. pulse and all. But there can be some other problems also because we are not giving it in the type it is. Like we are not giving ions and all. We are giving electrical currents there, charge there, right? So what is the primary concern addressed by stimulating thermal damage in brain tissue? So we are considering that there will be some heat being generated. So what will be effect of the heat on the brain tissue that we are stimulating? So is it to increase brain temperature for therapeutic purpose, to minimize tissue damage during surgery, to enhance cognitive function, to prevent mobile phone radiation effects?
I think it is B, sir. Minimize tissue damage during surgery.
Yes. So simulating thermal damage in brain tissue aims to understand the extent tissue will get damaged by procedures such as removing the tumor. In, okay. This thus ensuring that damage is limited to the targeted region and minimizing adverse effect of surrounding healthy tissues. Okay, then comes the thing that you were discussing. What is the significance of meshing in COMSOL multiphysics simulation? It determines the color scheme for visualization. It enhances the aesthetics of the simulation. It is crucial for solving equation in solvers. Helps in defining boundary conditions
It is crucial for solving equation it solvers.
in solvers because we cannot solve the equation for each and every point, and that also in detail we don't know how many points there are. So we are generally creating mesh and we are solving equations only at the crossing points, and we are assuming that this is the average of this, or if this is similar it is representing the whole area. And we have different meshing for different areas. The areas which are more critical, or we are going in details in that area, we have closed mesh, very close meshing, where we have very similar kind of structure. There we can go for some less dense meshing and all. Okay, so it is crucial for solving equations in solver
Yes,
to
sir.
obtain simulation results accuracy. Since you work on COMSOL, would you like to add something on this?
No, sir. I am working in CFD only. So we use
Okay,
create mesh and solve it. So the
okay.
same thing only they are doing. They are solving partial differential equation. For that, they need some
Yes.
boundary condition inside domain. They don't know that we will get from the solution.
Mhm. And in your case also, is it having different mesh density according to the
Yeah,
area
mesh
that?
is big mesh, maybe millions or may
Yeah,
go to billion also. So we have a
yes,
big HPC to solve that big supercomputer.
yes.
So
Yeah, I did a small part of it during my master's project. There also we were creating for one single device. I did not that complex. There also we used to do meshing and all, and we were solving this. That was for device level simulation. I think very similar to yours. Yours is thermal, right? You are doing thermal simulation. CFT is thermal,
no,
right?
sir, water, naval CFD, water, external
Okay,
flow,
what
ships and
channel flow?
submarine.
Okay, okay.
Ah, yes,
We
yes,
were
similar
doing
to that.
this current and charge simulation and all. Okay, so there also same kind of messing and all. It is a standard. We can say that we are not able to solve everything, so we are creating mess and all. So this is something very common in all the simulation tools we have. So next is how can animations of simulation result be saved for presentation? By exporting as an image file, by directly embedding it into PowerPoint, by converting it to a GIF format, or by saving as a video file.
I think simulation animations can be video file you can or directly PowerPoint is showing directly PowerPoint.
Uh, we can show uh, directly in PowerPoint, but that will again, like, there is no actually direct interfacing, we can say. So best will be to save as a video file, right? So that we can then we can embed it into a PowerPoint. This I just kept because this is something that was being discussed in the lecture. Okay, so it is by saving as a video file. Then something you talked about, boundary condition. So which boundary condition is applied to simulate electrical simulation, electrical potential, floating potential, terminal, or boundary current source.
He is saying terminal is most appropriate.
Yeah. And why is it so? Because in the context of simulating electrical simulation using COMSOL, the boundary conditions applied to simulate electrical simulation is typically referred to as terminal boundary condition. This allows for an specification of electric potential at particular region of area, which is like in all these generally boundary conditions are kept because in the bulk we can say that they are being modeled by a similar property or similar equation. But on the terminals there will be some different equation in play, right? So in this case it is the terminal boundary condition that is of relevance. Fine. So that's all for today. Any questions?
No, sir. I think the small session only. I covered this time, sir. 4 or 5 lectures
Yeah,
were there. Because last 2 times I said askew, so
yeah, yeah,
I.
yeah. Some part was
uh,
missing, not an issue. Yeah,
right. Okay. So I covered
yeah.
it. Now next is auditory thing. Next
Auditory, yeah.
week.
Next
Mm.
lecture, yeah. And
Okay, sir.
mostly
I'll
like
wait.
yeah. Now I am putting the summary at the beginning so that even if something is not covered, some part will get covered there. Though in details you can cover, like you certainly have to cover in the lecture, right? So that's all for today. See you in the next class.
Thank you, sir. Thank you very much.